Just give me a minute. All right, so this is the chapter we're going to start with. We are starting with the cell cycle and cell division. So ये chapter आज हमारा हम करेंगे यहाँ पे start. So let's see. इसमें आज हमें क्या देखना है. तो first of all, you have to know that from this chapter you get to see around like 1 to 2 questions. So 1 to 2 questions is something you will usually find coming from this particular chapter, and the same thing also happened yesterday also. So in the last the yesterday's NEET exam, what we got to see that you have like 2 questions coming from this particular chapter. So please remember you get to have 1 to 2 questions coming from this particular chapter of cell cycle and cell division. Let's not forget this chapter is your chapter number 10. So we also call it as a B10, right? Biology chapter number 10. So that's a cell cycle and cell division. So that is something we'll get to, right? Anji beta bataiye. Someone has raised their hand. Yeah. Acha, someone has dropped me a message. Yeah. Wait a minute. More weightage, I think roughly you get to have like 2 questions, Krishna. Roughly you get to have a 2 weightage. It is going to vary from year to year, right? Roughly you get to have like 2 questions from this particular chapter. At times you might find someday they might ask you 3 questions, but generally you don't go beyond that. Okay. Now, and the questions are also going to be like quite simple over here, so you don't get to have any issues while solving those questions. So now let's have a look over here. So in this chapter, we'll be discussing something in terms of the mitosis, meiosis. That is something we are going to discuss over here, and then the cell cycle as well along with that. So when you discuss a cell cycle, when you start with this topic of your cell cycle, the very first thing you have to understand, the cell cycle itself is done into like 2 parts. This has been done into 2 parts. You will have something like your interphase, and then you have M phase. So that is the 2 things you have to know, that your cell cycle itself is divided into 2 parts. You have an interphase and you have an M phase. ये 2 चीजें आपको यहाँ पे देखने के लिए आपको मिल जाती हैं यहाँ पे, right? Now there is one thing you have to understand, that all the cells undergo division, right? Majority of all the cells undergo division. There are going to be few cells which do not undergo cell division. So like for example, you can talk about something like a neuron. So neuron is a cell which won't undergo division once it has been formed. एक बार आपका यहाँ पे न्यूरॉन बन गया, देन उसके बाद आपका सेल डिवीजन आपका पे नहीं होता. इट वोंट बी फॉर्मिंग न्यू सेल्स ओवर देयर, राइट? सिमिलरली आई विल फाइंडिंग इवन योर कार्डियक सेल्स. कार्डियक सेल्स जो आपके होते हैं यहाँ पे, द कार्डियक सेल्स व्हिच यू हैव, they also do not undergo division. Once they have been formed, they do not regenerate themselves. So अगर कार्डियक सेल्स को कुछ हुआ, हार्ट के सेल्स, अगर हार्ट के सेल्स को अगर कुछ होता है, then you cannot replace them, right? So there are going to be few cells which once they have been formed, they do not show the tendency of one more cell division, right? So neurons are the example, cardiac cells are some of the example over here, right? But other than that, you get to see majority of the cell will have this potential to undergo division. They can undergo division and they can always divide. They can replace the old cells. They can replace the slogged off cells. They can always ensure that the new cells have been formed. So cell division is something which will happen throughout your life. Will just keep on happening. So you have an interphase and you have an M phase over here. Now, initially we'll be discussing something in terms of the interphase. Now, interphase is a phase where you will find the cell is going to prepare itself for possible division. ये आपका वो टाइम होता है जब सेल अपने आप को तैयार कर रहा होता है फॉर पॉसिबल डिवीजन के लिए. सो वी आल्सो कॉल इट लाइक प्रिपरेटरी स्टेज, या मैं कह सकता हूँ. तो प्रिपरेशन आपकी यहाँ पे इस स्टेज के अंदर की जाएगी. सो यू विल बी प्रिपेयरिंग योरसेल्फ for the possible division, preparation for division. That is something I'll be finding happening in my interface. Okay. Now let's try to understand what will be happening in this specific interface. Let's try to see what are the events which have been happening over here. So first of all, let's see the basic idea about the cell division. Cell division is a very important process in all living organisms. During this division, डीएनए रेप्लिकेशन एंड द सेल ग्रोथ ऑल्सो टेक्स प्लेस. तो आपका डीएनए रेप्लिकेशन भी यहाँ पे आपका होगा और आपके यहाँ पे सेल ग्रोथ भी आपको होते हुए आपको मिलेगा. दीज आर द टू थिंग्स व्हिच यू विल बी फाइंडिंग हैपनिंग ओवर हियर, राइट? All this process, that is cell division, DNA replication, and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells. There's one thing you have to understand very clearly, that whenever the cell division happens, so what is actually happening? You have a single cell, and from that cell, finding one more cell is getting formed. So when this division is happening, it has to take place in a very coordinated manner. इसको ये जब आपको करना है, ये आपको बहुत तरीके से आपको इसे करना है, बहुत सही तरीके से आपको इसे करना है, ताकि जो भी चीज यहाँ पे हो, whatever has been formed over here has to be formed in a very correct way. It has to be formed with no errors over there, right? So that's why it has been said it has to be formed in a very coordinated way, so that you get a right DNA formed and right DNA should be sent into the new daughter cell. So that is something which has been mentioned over here, right? The stages through which cell passes from one division to the next is called as my cell cycle. So these stages are been called as my cell cycle. Okay, now let's try to understand. So these are my cell cycle. Here you can get to see the cell cycle. So in the cell cycle, you will have something like your G1 phase, right? Then you have a M phase, sorry, then you have a S phase. So this will go in this particular direction. Then you will be having your S phase. After S phase, you will have your G2 phase. And after G2 phase, you will have the M phase, where the actual division will happen, right? Where the mitosis will take place over there. Now, there's one thing you have to understand. Whenever we discuss something about the interphase, now, please know that very carefully, that interphase is going to take around 95% of the time of the cell cycle. So interphase जो आपका यहाँ पे होता है, ये आपका बहुत ज़्यादा time consume करता है. So basically, cell is going to prepare itself for a possible division, and that much amount of time is being taken by my interphase. I would say rather the most amount of time is being taken by the interphase. So 95% of the time of the cell cycle is being occupied by my interphase. So this is also one thing which you have to be very much clear with, that this is what interphase is going to occupy. So the real division, the division which is actually happening is only taking around 5%. तो जहां पे आपका डिवीजन हो रहा होता है इन अ रियल सेंस, वो सिर्फ आपका 5% ही वहां पे आपका यूज करता है, ऑक्यूपाई कर रहा है आपका वहां पे सेल साइकिल का. ठीक है, तो ये बात आपको यहाँ पे, दिस इज वन थिंग यू हैव टू रिमेंबर. Now let's slowly understand what are these phases, what are this G1, S phase, G2 phase, and what are the events which have been happening over here. Now, there's one thing should be you all should be very much clear with, that you will find, you will be finding that G1 phase, S phase, and G2 phase are the one which become a part of my interphase. So you have a G1 phase, you have a S phase, right? You have a S phase, and then you have a G2 phase. These phases are collectively a part of my interface. They all become a part of my interface. कुछ issue हो रहा है यहाँ पे, just wait a minute. Some So G1 phase, S phase, and G2 phase are going to be a part of my. Okay, someone has a question. Josh has a question. Josh, sir, should I leave the class? I'm thinking the topic you're going to talk is no, beta. So that is the thing. This batch actually goes slightly like it's behind from the normal batch which are happening from the 5:30 class. So yes, ये जो हम कर रहे हैं यहाँ पे, that is something you have already done. So that's why I told you. This is something you might have been there in the class. हाँ, ठीक है. In that case, yeah, you can leave. Huh? Yeah, no issues, because it will get, you will be spending your time over here. Okay, solve some questions. Yeah, chalo, bye. Okay, so there's something you have to know. So you have a G1 phase, S phase, and G2 phase, and that is something collectively is a part of my interface, right? Now, let's try to understand first of all my G1 phase. So let's see what is the story of your G1 phase. So when we discuss our G1 phase, let's see what are the events that have been happening over here. So let's see, we have a G1 phase. Now, when you discuss a G1 phase, now please understand one thing. This is the first phase of my interphase. Interphase का सबसे पहला जो मेरा phase होता है वो मेरा होता है यहाँ पे G1 phase. Now, G1 phase is basically known for high amount of protein synthesis right? And also cytoplasmic growth. So there are a few things you have to know over here. You will find protein synthesis taking place over here. We'll have protein synthesis happening over here. That's one thing you have to know. कि यहाँ पे आपका protein synthesis एक काफी अच्छे amount में आपको यहाँ पे होते हुए मिलेगा, right? Then you will have the second thing which will be happening here is that is going to be your cell organelle duplication. Cell organelle duplication. Now let's understand what do we mean by cell duplication. Krishna, you can stay till you have your battery in your like phone now. Till then you can stay. Once it's not there, by default you'll be leaving the class, right? Okay. So you'll have a cell organelle duplication. It's also something which will be happening in my G1 phase. That's the second thing you have to know. Now, when we say cell organelle duplication, that means what? कि आपकी जितने भी organelle है, like mitochondria, Golgi apparatus, you have your ER, lysosomes, they will undergo duplication. So you have to make their copy as well. So that will be happening in my G1 phase. So G1 phase is a phase in which you'll get to see that all my cell organelles, which are going to be like mitochondria, chloroplast, all those organelles, will duplicate themselves. Because if the possible cell division happens, then you need to have a new cell should also have all those organelles with itself, right? So that's why this duplication will happen in the G1 phase. So that's the second thing you have to know. The third thing you have to know that you will find cytoplasmic growth keeps on happening. Cytoplasmic growth is very like something which happens throughout the cell cycle, right? So we'll have a cytoplasmic growth. Cytoplasmic growth will be seen in the G1 phase. So that is something you keep on finding that cytoplasmic growth आपको यहाँ पे होते हुए आपको यहाँ पे मिल जाती है, right? So these are the three main events you should know over here happening. Along with that, you also find RNA synthesis also taking place. That happens in G1 phase as well as in my G2 phase. हर जगह आपका RNA synthesis तो आपका होता ही है. But ये तीन चीजें आपको यहाँ पे याद रखनी है. These three things, first of all, you have to very clearly know these are the events happening in the G1 phase, right? Now, if they ask you in the interphase, you have a G1 phase, S phase, and you have a G2 phase. Which phase will be the longest one? Then your answer is going to be my G1 phase. So of these three, G1 phase is the one which takes a much more amount of time compared to the remaining two. ठीक है? तो ये बात आप यहाँ पे याद रखना. So that is what we will be finding happening in the G1 phase. After that, you will be having your S phase. तो ये तो हमने देख लिया G1 phase में और यहाँ पे क्या हो रहा है. इसके बाद मेरा यहाँ पे आएगा S phase. So now after this, I'll be having my S phase over here. So let's see S phase में मेरा यहाँ पे क्या होता है. So S phase is a synthesis phase. And this phase is mainly known for my DNA replication. That is a major event which I'll be finding happening over here is my DNA replication. Only 10%. All right, Krishna, it's your call. So yeah. Okay, in case if you happen to get your electricity back, then you can join in again. Right, so now you have a S phase over here, and the S phase will be known for my DNA replication. That's one thing you have to know. It is been known for my DNA replication. एक ये चीज आपकी यहाँ पे हो जाएगी, right? So DNA replication is one of the very important event. So you have your DNA, right, and you have to make a copy of a DNA. So one more copy will be formed over there, right? So DNA replication is something one of the very important event of my cell cycle, and that is happening in the S phase, right? Now there's one thing you should very clearly know. During the S phase, the DNA will get doubled. During S phase, आपका DNA यहाँ पे double हो जाता है. So DNA will get doubled over here. So DNA gets doubled, right? So DNA gets doubled over here. That means what? If let's say initially you have a 2C amount of DNA, by the end of the S phase, you will have a 4C DNA.
4C.
Right, you have a 4C DNA. So DNA will get doubled over there suddenly, right? But the number of chromosomes remain the same. ये बात रखना, number of chromosomes remains the same. We will find number of chromosomes remains the same. Remains same. So chromosome number यहाँ पे change नहीं होगा. If initial chromosome number is 23, if initial chromosome number is your 23 pair, then after DNA replication, the chromosome number is going to remain the same. So that should be clear to all of you that chromosome number is not going to change, but yes, DNA will get doubled. DNA का amount आपका double हो जाएगा यहाँ पे, right? So that is the one of the important event happening in the S phase. That is going to be my DNA replication. Now, along with that, couple of more things will happen. So we have seen the S phase. क्या हमने यहाँ पे देखा कि यहाँ पे क्या हो रहा है? But let's see some more things about the S phase, right? So let's see over here. So S phase is also known for a couple of more things. So we have seen DNA replication, but along with that, it is also known for centrioles duplication. Centrioles duplication. So centrioles will be there in the cytoplasm, and they also undergo duplication. So their number is also going to increase over here, and that is also something happening during the S phase. ये भी आपका S phase के अंदर ही होता है. Along with that, one more thing which will be happening over here, and that one more thing is going to be your S phase is a time where you will also find that your histone proteins are been formed. Your histone proteins are formed. Histone proteins are formed. That is also one thing you have to remember, that histone proteins have been formed over here, right? So central duplication will be finding happening over here. Histone proteins will be formed over here. And the last thing which we have already discussed is going to be your DNA replication. Along with this, you'll have your DNA replication will be happening in this stage. This is what my S phase is known for. And it is a very critical stage actually, because you have to carry out all those events very precisely. If you don't do them precisely, then that is not something which is going to be like helpful for the cell division. Okay, तो ये तीन चीजें आपको यहाँ पे याद रखनी है. That is all happening in the S phase right after the G1 phase. Now after that, which phase it is going to be? क्या होगा इसके बाद मेरा next phase कौन सा होगा? G2 phase. G2 phase. Now the next phase is going to be G2 phase. Now before that, let's have a look what they have written. In the interphase, cell grows in size, prepare itself for the next division. Interphase is the most active phase of the cell cycle. Bilkul, number of things are happening over here. Protein are getting formed, RNA is getting formed, cell division is your DNA replication is taking place. All those events are happening. It is a significant to note that 24-hour average duration of cell cycle of the human cell. Cell division proper lasts only for about 1 hour. So we can say that you have a cell cycle which lasts for my 24 hours. एक ह्यूमन सेल का सेल साइकिल होता है 24 घंटे का, 24 आवर्स का, राइट? नाउ of this 24 hours you will get to see that 23 hours are been spent in my interphase. This much amount of time is been spent in your interphase, and it only takes around like 1 hour. It only takes around like 1 hour for your mitosis or M phase. So that is quite interesting, that majority of the time is been spent in your interphase and a small amount of time is spent in the M phase, right? The interphase lasts for more than 95% of the duration of the cell cycle, something we have already seen. Right? So that is something you can get to see over here. There is a cell cycle, which we'll be discussing again later on. So now let's have a look at the G2 phase. So now when we discuss a G2 phase will be having certain things happening. Like G2 phase is known for, again, for my protein synthesis.
RNA.
Right. Protein synthesis आपका यहाँ पे होता है, आपको मिल जाएगा. You will have your RNA synthesis taking place over here. You'll have your RNA synthesis taking place. Right. And the third thing is going to be your cytoplasmic growth. वो तो आपकी होती रहती है. Cell तो आपको grow करता ही रहता है. So cell will just keep on growing at this point of time as well. Cytoplasmic growth. So these are the few things you can find happening in my G2 phase as well. Right. So G2 and G1 are like very similar. Like the only thing is in G1 specific is going to be that G1 will be having cell organelle duplication happening over there. something you don't find happening in the G2 phase, right? So that is something you will find happening in my G1S and G2 phase, and then you have the M phase where you actually find the mitosis will takes place. तो ये हमने यहाँ पे देख लिया. That is something we have seen. यहाँ तक सबको क्लियर है? I hope यहाँ तक किसी को कोई डिस्टर्बेंस नहीं है. I hope this is something clear to all of you.
Yes, sir.
Yes, sir.
पूछ लेते बाकी सबसे. We have what? महालक्ष्मी लेट हो गया आज क्लास में आप. शिखा तो है पहले से. Yeah. Saurav, I hope it's clear to you as well. Saurav, Aisha, then we have Mantasha. I hope it's clear to you as well, and Aditya. All right. Aditya, are you able to hear us?
Yes, sir.
All right. Aditya, tell me something from where you are actually. From Karnataka, from where you are?
Yes, sir. I am from Karnataka, Hubli.
And from Karnataka, like within Karnataka, from where?
Hubli, sir. Hubli.
Ah, Hubli. Hubli,
Yes, sir.
Dharwad. Okay. Cool. Nice. Right. Okay. Chalo. So that is something we'll get to see over here. So that is something we'll find in my G1 phase, G2 phase, and my S phase, right? So these are the cell, these are the three gaps. These are the interfaces you'll get to see. Now, let's talk about these phases quickly over here. There are some statements written over here which I have already discussed. G1 phase is a cell metabolically active, continuously grows but does not replicate its DNA. All right. G1 phase is a period where in which the cell grows and carries out normal metabolism. Most of the organelle duplication occurs in this phase. So majority of the organelle duplication is something you'll find happening in this particular phase. During G1, cell rapidly synthesizes types of RNA and protein. यहाँ पे आपका प्रोटीन सिंथेसिस भी आपके यहाँ पे हो रहा है और यहाँ पे आपका आरएनए सिंथेसिस भी आपके यहाँ पे हो रहा होता है. Then replication of nuclear DNA, synthesis of histone protein takes place in the S phase. So this is a time where we will find the nuclear DNA will get formed, your histone proteins are getting formed in the S phase. During this time, the amount of DNA per is going to get double. So as I've told you, the amount of DNA will get doubled over here. So if the initial amount is going to be my 2C, the amount becomes 4C over here. However, there is no increase in the chromosome number. The chromosome number is not going to change. ये बात भूलना मत. तो अगर मैं आपके सामने कोई एनिमल रखता हूं, if I give you some animal and animal has 12 chromosomes, then after S phase, how many chromosomes the animal will be having?
12 chromosomes.
12 chromosomes.
12 ही रहेंगे. That is not going to change, right? And you will find such question coming in exam, so please make sure you don't miss out on them. Number of chromosomes are not going to change, but the amount of DNA will get doubled. In animal cell during S phase, DNA replication begins in the nucleus and central duplication in the cytoplasm. So that is also something which will be happening, right? Central duplication. And then you have a G2 phase, protein synthesis will take place. It is preparing itself for the possible division. So in G2 phase, your cell is actually ready to carry out the mitosis. G2 phase is a period of cytoplasmic growth. ये आपके cytoplasmic growth का ये period आपके यहाँ पे आ जाता है. So these are the things you know. Now let me tell you something which is going to be slightly different. Let me make a cell cycle. Let me make a circle to make you understand a cell cycle. So let's say this is my M phase. This is my M phase. Something which you are not concerned about right now. Gayatri, I hope it's clear to you as well. मैं भूल गया पूछना, Gayatri.
Yes, sir.
ठीक है. Then let's assume you have your G1 phase. And this is my G1 phase. After G1 phase, let's see you have S phase over here. यहाँ से चालू हो रहा है मेरा S phase. Now, something I'm going to tell you right now is something which in NCERT they haven't explained that in detail, but we have seen question coming from this particular part as well in the past. So that's why we have to focus over here. And then the last phase is going to be your G2 phase. This is my G2 phase. Okay. Now, so you have a G1 phase, you have a S phase, and you have a G2 phase. Now let's have a look what will be happening over here. Now, G1 phase is something you're all familiar with what actually happens. So we don't have to get into details of that. Here we are going to discuss something known as my checkpoints. यहाँ पे हम कुछ discuss करेंगे and that is called as my checkpoint. So between the G1 and S phase, there is something known as my checkpoint will be seen over here. And between the G1 and S phase, the checkpoint is going to be my checkpoint number 1. So सबसे पहला जो checkpoint हम यहाँ पे बात करेंगे, that is going to be the checkpoint number 1. And checkpoint number 1 आपका जो यहाँ पे आएगा, that will be seen between my G1 and S phase. So let me see. So you have checkpoint number 1. Okay, you have a checkpoint number 1, and that checkpoint number 1 comes between my G1 and S phase. Now let's try to understand what this checkpoint is known for. What are these checkpoints and what they are actually going to do over here? See, now let's understand. I'll give you a general idea about the checkpoints, right? Now, what do you get to see now? Assume that you are traveling from Karnataka to Mumbai, right? From the highway by road. So what will happen? You will have some checkpoints, right? You get to have a checkpoint where you have to pay the toll, right? वो आपका toll नाका आता है, you have to pay the toll and then
Yes, sir.
या तुम हाईवे पे तुम अपनी गाड़ी भगा सकते हो, ठीक है? इस एग्जांपल को थोड़ा हम यहाँ पे चेंज कर देते हैं. Assume that you are like crossing a boundary. You are crossing an international boundary over here. Assume that you are traveling from like Punjab and then you are traveling towards Pakistan, right? तुम boundary cross कर रहे हो बाइक पे. समझ लो तुम 5-6 जन वैकेशन पे बाइक पे जा रहे हो घूमने के लिए वहां पे, right? So what will be happening over here? Now tell me something. If you are trying to cross an international boundary, then what are the things they are going to check from you? What are the things they are going to like ask you at the checkpoint? Checkpoint पे क्या देखा जाएगा? बताइए महालक्ष्मी, क्या देखा जाएगा checkpoint के ऊपर?
Sir, they take information that where we are going.
ठीक है, एक तो ये पूछा जाएगा कि भैया कहाँ जा रहे हो? नीट की एग्जाम की तैयारी नहीं करनी. Along with that, anything else, महालक्ष्मी, और कुछ?
So where are they
Sir,
coming from? For what reason? They ask.
हाँ, they will ask you यार क्यों जा रहे हो? हाँ ठीक है, right? Where you are going? Why are you going? Okay, anything else, Saurav, और कुछ?
सर इक्विपमेंट्स भी चेक करते हैं, लाइक कोई डेंजरस इक्विपमेंट्स लेकर नहीं जा रहे.
हाँ, ये बात भी सही है शिखा कि आप चेक कर लोगे वहां पे कि आपके इक्विपमेंट्स मतलब जो भी आप लेके जा रहे हो वो सब लीगल है, कुछ इलीगल आप लेके नहीं जा रहे जिससे आप किसी को डैमेज कर सको, राइट? सो ऑल दोस थिंग्स हैव बीन डन एट अ चेक पॉइंट. उसके साथ साथ आपका पासपोर्ट चेक किया जाएगा, वीजा चेक किया जाएगा, ड्राइविंग लाइसेंस चेक किया जाएगा whether you have or not. If everything is falling in place, then they will allow you to go. अगर सब कुछ सही रहता है then they will say कि अच्छा ठीक है सब कुछ सही है यहाँ पे, now let the person pass and let the person get into the Pakistan. So वो आपको वहाँ पे करने देंगे, right? Similarly यहाँ पे checkpoint इस तरीके से आप यहाँ पे काम करेगा. So you have a checkpoint number 1. Checkpoint 1 is something which will have a role to ensure that or check that all the things are clear or all the things are in an appropriate way so that we can get into the S phase. So ये जो checkpoint number 1 मेरा यहाँ पे होता है, यहाँ पे क्या चेक किया जाता है? यहाँ पे आप ये चेक करते हो कि G1 phase के अंदर right amount of protein बने हैं या नहीं बने हैं. So याद रखना, in the checkpoint number 1 is you are going to check that whether you have appropriate amount of protein synthesis or not. So this is something should be clear to you. So in checkpoint number 1, you are going to check appropriate amount of protein synthesis has taken place or not. तो वहां पे वो चेक करेगा कि क्या सही अमाउंट में यहाँ पे प्रोटीन बना है या नहीं बना है जीवन फेज के अंदर. अगर सही अमाउंट में यहाँ पे प्रोटीन नहीं बना है, अगर सही अमाउंट में अगर आप यहाँ पे प्रोटीन नहीं बना है, then what will happen? The cell cycle will not move into the S phase. Cell cycle will not move into the S phase because you don't have a right amount of protein which should be there in the G1 phase. So that should be clear to all of you that in G1 phase, if you don't have a right amount of protein formation, then you cannot allow the cell cycle to move into the S phase. So that is something done by my checkpoint number 1. So checkpoint number 1 यहाँ पे ये चेक करेगा, अगर प्रोटीन बने हुए कोई दिक्कत नहीं है, you get into the S phase. अगर प्रोटीन बने हुए आपके पास यहाँ पे नहीं है, then वो तब तक सेल साइकिल आगे बढ़ेगा नहीं जब तक सही अमाउंट में यहाँ पे प्रोटीन नहीं बन जाता. Is this clear to all of you?
यस सर.
ठीक है,
Yes, sir.
all right. So this should be clear to all of you. Yeah. If anyone has a question, yeah, महालक्ष्मी बताइए.
सर चेक पॉइंट को स्पेसिफिक नेम नहीं है क्या सर?
चेक पॉइंट को चेक पॉइंट बोलेंगे. चेक पॉइंट का अंदर कुछ प्रोटीन, यहाँ पे कुछ प्रोटीन इन्वॉल्व होते हैं. मतलब अगर तुम बॉर्डर क्रॉस करते हो वहां पे जैसे गार्ड्स वहां पे खड़े होते हैं ना, यहाँ पे आपके कुछ प्रोटीन इन्वॉल्व होते हैं यहाँ पे,
Yes, sir.
राइट. तो प्रोटीन का नाम अभी हम नहीं देखेंगे, वो बाद में देखेंगे हम लोग. ठीक है, अभी हम इनका रोल यहाँ पे देख लेते हैं, बिकॉज वो प्रोटीन का नाम कभी एग्जाम में आएगा नहीं, बहुत अनलाइकली है, इसलिए हम उसे बाद में देखेंगे, राइट.
Okay.
कॉल मतलब साइक्लिन एंड सीडीके, बट वो हम बाद में देखेंगे. अभी हम इनको रोल देख लेते हैं यहाँ पे, राइट. सो दैट इज माय चेक पॉइंट नंबर 1 ओवर हियर. So after checkpoint number 1, now we'll have to move on to the another checkpoint. Now the next checkpoint is going to be between the G2 and M phase. This is my checkpoint number 2. This is going to be my checkpoint number 2. And then you have one more checkpoint that is going to be between the mitosis. Yeah, महालक्ष्मी बताइए.
एस फेस और G2
कोई
फेस
checkpoint नहीं होता. ना कोई checkpoint नहीं होता. कोई checkpoint नहीं है. जरूरत नहीं है वहां पे चेक पॉइंट की. इसलिए हमने एक मेन यहाँ पे गेट लगा दिया ना, माइटोस में जाने के लिए हमने एक बड़ा चेक पॉइंट यहाँ पे लगा दिया, तो यहाँ पे रखने की जरूरत ही नहीं है. वैसा है. ऐसा समझो यहाँ पे एस फेस से G2 फेस जाना मतलब इट्स लाइक मूविंग फ्रॉम इस्लामाबाद टू रावलपिंडी, राइट. नो नीड ऑफ एनी काइंड ऑफ चेक पॉइंट. कोई देख नहीं रहा, जाओ जहाँ जाना है जाओ, राइट. बट अगर आप वहां से आप क्रॉस करते हो बॉर्डर आगे और आगे जा रहे हो, देन वहां पे चेक पॉइंट आ रहा है. ठीक है. सो यू हैव अ चेक पॉइंट नंबर 3 ओवर हियर एज वेल, व्हिच इज गोइंग टू बी between the M phase. So now let's jump on to my checkpoint number 2. Let's try to talk about my checkpoint number 2 over here. Wait a minute. By the way, कैसी है गर्मी आपके वहां पे? People from South India, Gayatri, Aditya and Mahalakshmi.
38 degrees sir.
बहुत है सर.
38 तो अच्छा है यार. 38 is still better.
सर
But
धारवाड़
sir,
थोड़ा
like,
बहुत कम है सर अभी.
हाँ.
but I was on like other cities sir, वहाँ 42 था कलबुर्गी.
हम्पी है ना सर, वही उधर सभी
Like
बहुत है सर.
yeah, it's गर्म
So
है.
but Gulbarga is one of the most like temperature ज़्यादा रहता है ना sir.
Okay, Gulbarga is something which will come in the northern part ना, एकदम northern part of Karnataka ना.
हाँ थोड़ा, लेकिन मैं वहाँ गया था सर like recently, इसलिए बहुत ज़्यादा था सर, बाहर नहीं निकल पाए.
Like because we have 40 42, how can you can have 38? That's what I'm thinking. But अच्छा यार. See, I do think that because Hubli and Dharwad are closer to Belgaum and some kind of, it is I guess slightly closer to Western Ghats, I would say. Maybe because of that you don't have that much amount of temperature. Maybe.
Yes sir.
थोड़ा बहुत रहता है सर धारवाड़ में, तो इतना ही हमारे को बस हो जाता है सर.
हाँ सर,
गर्मी.
पहला मतलब उतना नहीं था ना सर, अभी डायरेक्टली इतना गर्मी थोड़ा सह नहीं पाते सर.
क्या मतलब क्या? हाँ, धारवाड़ इसमें ज्यादा डिफरेंस है. 30 किलोमीटर डिफरेंस ना ओनली यू हैव धारवाड़ हुबली.
Yes sir, 30 kilometers.
So सही है. But yeah, you can visit na, like you people must be having Amboli closer to you na, comparatively.
Yes sir.
Make a visit in monsoon. All right, chalo. So now what will be happening here? Yeah, checkpoint number 2. So in the checkpoint number 2, what you get to see now, this is something which has been coming between your G2 and M phase के बीच में, right? This is what you will get to see. This is what checkpoint number 2 is going to be known for. Right. Now what you have to understand here is what this checkpoint is known for. Now let's please understand this thing, that your DNA replication will take place in the S phase. Now DNA replication should take place very precisely. DNA replication should always takes place very precisely without any errors. बिना कोई error या बिना कोई concern के replication आपका अच्छे से होना चाहिए. Replication should take place very precisely. Right. Now what checkpoint number 2 is going to check now, whether the DNA replication has taken place precisely or not. So it is going to scan the DNA and will look for the errors. So it is going to check the DNA replication. So checkpoint number 2 is going to check the DNA replication checking. So DNA replication की चेकिंग यहाँ पे की जा सकती है. राइट. सो डीएनए रेप्लिकेशन की चेकिंग आप करते हो यहाँ पे चेक पॉइंट नंबर 2. बेसिकली आपका जो डीएनए बन चुका है उसे आप देखते हो ध्यान से कि सही से बनाया नहीं बनाया, राइट. नाउ लेट्स अज्यूम दैट यू आर डूइंग द चेकिंग ऑफ द डीएनए रेप्लिकेशन, राइट. सो आप अपना जो डीएनए बनाए यहाँ पे उसे आप स्कैन कर रहे हो, and you get to realize the checkpoint number 2 get to realize that there are some errors. So assume that while checking you realize that you have some error over here, right. Now if you get to see some error in the DNA, what will now happen? Now if you get to see error in the DNA, now you will initiate a DNA repair mechanism. You will initiate a DNA repair mechanism. DNA repair. अगर डीएनए में कोई दिक्कत आती है यहाँ पे, देन आपको करना कुछ नहीं है यहाँ पे, आप अपना चालू कर दो आपका डीएनए रिपेयर यहाँ पे, राइट. दैट इज समथिंग इट इज गोइंग टू स्टार्ट कि अच्छा कुछ एरर है यहाँ पे, यू स्टार्ट द डीएनए रिपेयर, राइट. नाउ व्हाट विल हैपन आफ्टर डीएनए रिपेयर, इट इज अगेन गोइंग टू चेक. एक बार डीएनए रिपेयर हो गया तो आप उसे वापस चेक करोगे कि अब सब सही हुआ या नहीं हुआ. तो समझो डीएनए रिपेयर होने के बाद आपने वापस से चेक किया डीएनए को, ठीक है. आपके चेक पॉइंट ने डीएनए को वापस से यहाँ पे चेक किया, एंड चेक करने के बाद आपको क्या दिखा वहां पे कि सब कुछ आपको वहां पे सही है, एवरीथिंग इज गुड. सो आफ्टर चेकिंग व्हाट यू गेट टू सी दैट एवरीथिंग इज गुड. सो अज्यूम दैट एवरीथिंग इज गुड एंड यू गिव द, आप उसे परमिशन देते हो, यस सब कुछ सही है, आप आगे जा सकते हो. Now as soon as you give them a permission, then what will happen? Now your cell cycle will move into the M phase. Cell cycle will now move into the M phase. उसने देख लिया कि चलो डीएनए मेरा ठीक है, आपने रिपेयर कर दिया सही से. Now you can move into the S phase. So now that will be happening, right? So now you can move into the, यहाँ से आप जाओगे अपना सक्सेसफुली अपना यहाँ पे M phase के अंदर आप चले जाओगे. So from here we'll get into the M phase, right? Now assume that after DNA repair you make the check, and after checking you get to realize that it is a no. No मतलब अभी भी एरर ठीक नहीं हुआ है. आपने चेक किया और आपने देखा कि नहीं, जो प्रॉब्लम पहले थी वो अभी भी आपके वहां पे आपको दिख रही है. अब देखो यहाँ पे क्या होता है. If the problem still persists over here, then you have to know that the cell will prepare to kill itself. The cell will stop the idea of cell division. It won't go further for the cell division and will initiate a programmed cell death. अगर आपका डीएनए अगर आप रिपेयर नहीं कर पाते, then you will initiate the programmed cell death. You'll initiate a programmed cell death. That means cell will now prepare a process to kill itself. Why it has been doing that? Because you cannot afford to have an error DNA into a new cell. You cannot compromise with the quality of a DNA getting into a new cell, and that is why you have to kill your cell. सिंपल सी बात है, आप नए सेल के अंदर आपका गड़बड़ वाला डीएनए आप आगे भेज नहीं सकते, इसलिए आपको अपने सेल को यहाँ पे खत्म करना पड़ेगा. क्योंकि अगर एक गलत डीएनए आगे चला गया, the wrong DNA gets into a new cell, then all the new cells coming from that cell will be having the error DNA. So that is why if your DNA has some kind of damage which cannot be repaired, then you have to initiate a process to kill the cell. बिल्कुल सही तरीका है, इसे हम कहते हैं क्वालिटी कंट्रोल. ठीक है, दैट इज समथिंग कॉल्ड एज क्वालिटी कंट्रोल, राइट. एंड दैट हैज टू बी मेंटेन्ड ऑल द टाइम. Something which we also maintain in the food industry as well. If let's say Gayatri has a factory of gulab jamun selling factory, and if one of the can of gulab jamun has some what do it has some kind of leaf is there in the can, right? So she won't allow that thing to get into the market. No, it is going to discard that thing, right? Similarly, it's the same thing happens over here, right? If a DNA is getting repaired, then it's all good. If it is not being repaired, then you will find a program cell will take place over there, right? So that is what you have to know about the checkpoint number 2. Checkpoint number 2 is known for checking scanning the DNA, whether it has replicated correctly or not. That is what my checkpoint number 2 is known for, and it is a very important checkpoint. अगर यहाँ पे कोई गड़बड़ होती है, अगर ये checkpoint काम नहीं करता, then you will find your DNA will get into new cell, which is a error DNA, right? So that is something you have to know. So is this thing clear to all of you? Is the checkpoint number 2 clear to all of you?
Yes, sir.
Sir, can you please say one more time, like explain one more time?
So what will be happening now? So you have this checkpoint number 2. It is known to check whether the DNA is been correctly formed or not. Newly formed DNA is correctly formed or not, right? Now let's assume you are scanning the DNA and you come across an error. You saw that there are some error in the DNA. So what the checkpoint number 2 is going to tell you? Checkpoint number 2 is going to tell you that you initiated DNA repair mechanism. वो क्या बोलेगा कि आप DNA repair mechanism को आप initiate करो. That means what? You try to repair the DNA. You try to ensure that the error you have in the DNA can be like brought into a normal condition or can be repaired. So that is going to, it is going to initiate, right? So now what the checkpoint number 2 has done, it has initiated DNA repair mechanism. It has initiated DNA repair. So after DNA repair, the checkpoint number 2 is also going to check again. Checkpoint number 2 क्या करेगा? बोलेगा कि आप repair कर लो. You start repair process, right? And after getting repaired, it is again going to check the DNA. Now during checking the DNA, if it come across no error, आपने repair कर लिया, आपने देखा उसे check किया, and you have seen that there is no error. Yes means it is all good. If it is all good, then you get into M phase. No issues. The issue has been resolved, and now you can get into the M phase. If the error is not repaired, even after trying to repair, if the error is not repaired, then it's a big no. Nahi hua repair. There is no repair which has taken place over there. In that case, cell will initiate a process of killing itself, which you call it as a programmed cell death. So this is where cell will say that, okay, we cannot allow this wrong DNA to get into a new cell. We have to kill the cell. This is what happens in the checkpoint number 2. So you should be very, very, very clear what is checkpoint number 2 is known for and why it is going to be so important. Okay? So I hope now it's clear to you, Aditya, and all the other people.
Yes, sir, it's clear.
Gayatri, Aisha, is it clear to you?
Yes, sir.
All right. And I think the same question goes for all of you. ठीक है, सौरभ, ठीक है यहाँ तक सब कुछ? All right. Okay. हाँ जी, महालक्ष्मी बताइए.
So how DNA get errors?
Sometimes happens during DNA replication. You can find कि if you study a biomolecular chapter, na, so you must have seen something in 11th. Yeah. So adenine के सामने क्या लगता है आपका? Thymine लगता है ना?
Yes, sir.
And guanine के सामने आपका लगता है आपका cytosine.
Cytosine.
Right? Sometimes what can happen ना, जल्दबाजी में what can happen ना, क्या adenine के सामने आपका लग सकता है आपका यहाँ पे cytosine. अगर ऐसा कुछ हो गया तो हो गया ना गड़बड़, हो गया error, right? Such error can happen. इसे कहते हैं mismatch based pairing. We call it as a mismatch based pairing. And let me tell you something, you have these errors and these errors are the only reason because of that sometimes the cancer happens. Right?
Okay.
So basically, if you study cancer in future in your college, they will initially teach you everything about the cell cycle. When your cell cycle stops working properly, that is one of the reasons for the cancer. Right? Your checkpoints, when they stop working properly, that can lead to a condition of your cancer.
Okay, sir.
These checkpoints are very important for regulation of the cell cycle.
Okay, sir.
They ensure that how the cell should be undertaking the division. ठीक है, चलो ये आपने यहाँ पे देख लिया. These are things you have seen. Then after you have a checkpoint number 3 जो हम अभी देखेंगे वो बाद में देखेंगे. Checkpoint 3 का काम आएगा बाद में, अभी नहीं. Right? Now let's see what has been written over here. Some cells do not exhibit cell division and they remain metabolically active. Right? So there are going to be some cells which are going to be metabolically active, but they do not show the cell division, like your heart cells. Then you will have some cells which will only divide as per the requirement. बाकी वो metabolically active रहेंगे. So such cells have gone into a G0 state. So let's see what has been written over there. Some cells in the adult animals do not appear to exhibit division, like your heart cells, and many other cells divide occasionally. Many other cells only divide occasionally. उसके अलावा वो लोग cell division show नहीं करते हैं, as they need to replace cells that have been lost because of an injury or a cell death, right? So if there are going to be some cells which will only divide when there's going to be some kind of cell death or there are going to be some kind of injury to that particular tissue, or else they won't divide. So such cells are in the G0 state. We call them as a G0 state. In this state, those cells are going to remain. So when you get a G0 state, now that means what? This is a state and in which you will find cell is going to be metabolically active. Cell आपका metabolically यहाँ पे active है, बस वो divide नहीं कर रहा, right? These cells do not divide further, exit the G1 phase and enter an inactive stage called as my quiescent stage. they get into an inactive stage called my quiescent stage of the cell cycle, which is also called as my G0 state, right? If the cell is in my G0 state, अगर मैं कहता हूं कि देखो ये cell है और ये G0 state में है, then that means what? The cell will be metabolically active, but it won't undergo any kind of division. It won't have any kind of intent to undergo division. So that should be clear to all of you. Simple thing. इसका आंसर देने के लिए आपको करना पड़ेगा अपना हैंड रेज. सो बताइए इसका आंसर यहाँ पे क्या होगा. बताइए गायत्री, क्या होगा इसका आंसर?
Sir, 95.
95 Gayatri. Seems like Shikha will also have the same answer over here and Aditya as well. So yes,
Yes,
90 is
sir.
going to be correct.
95.
Yeah, true. ठीक है. DNA synthesis takes place during. Again, you'll have to raise your hand to answer this. बताइए महालक्ष्मी. Mahalakshmi is not raising her hand. Mahalakshmi, why you take so much amount of time to raise your hand, to unmute yourself? क्यों ऐसा बेटा, क्यों? What did Aditya tell me?
So, yum.
Mahalakshmi said M phase, na? Mahalakshmi, minus 25. Minus 25 to you from me. अगले टेस्ट में इसे मैं काटूंगा मैं. अगले टेस्ट में आपके 100 आएंगे ना, तो उसमें मैं minus 25 कर लूंगा. इतना बताने के बाद भी M phase. आदित्य बताओ. Aditya, DNA synthesis takes place where? During?
Sir, yes, sir.
S phase. Right. Now, Gayatri, you have to tell me, or Mantasha, you tell me what are the other events happening in the S phase. Mantasha, the question is, in S phase, you find a DNA replication takes place. Along with that, what are the other things which have been happening over here?
Protein synthesis.
Yeah. You will find what kind of duplication will be happening over here.
Centriole duplication,
Central replication.
DNA duplication,
DNA replication.
and histone proteins formation.
Right. Bilkul sahi. Yeah, these are the things shall be finding. Histone duplication, central duplication, and my DNA तो बनी रहा है यहाँ पे. ठीक है, simple है, right? Give me this sequence. Now this question was also asked in the yesterday's exam. Now tell us what is the correct sequence of the cell cycle over here. Sorry sir, I cannot unmute myself. Okay. All right. So इसका आंसर यहाँ पे क्या हो जाएगा? सौरभ ट्राई करोगे यहाँ पे? गलत हुआ तो गलत हुआ. कौन इतना सोचता है? 1 2 3 1 2 3 4 कौन सा वाला बताओ?
3.
3 आयशा. महालक्ष्मी, क्या होगा इसका आंसर?
Sir, option D.
ऑप्शन डी या ऑप्शन 4 इज गोइंग टू करेक्ट आंसर बेटा. पहले आपका आएगा G1 फेज, देन यू हैव अ S फेज, देन यू हैव अ G2 फेज, एंड देन यू हैव अ माइटोसिस M फेज. ठीक है,
Mitosis.
तो ये आपका हो जाएगा यहाँ पे आपका सही वाला साइकिल. यस, ये क्वेश्चन आंसर आएगा यहाँ पे आपका ऑप्शन 4.
So, +4.
+4. हाँ, +4. यार राइट, ठीक है. +4 करके आपके 25 कम करेंगे. ओके, देन क्वेश्चन नंबर 1 over here. Now you have to read the statement thoroughly and then you have to tell me which of them is going to be a correct statement for my G1 phase. खुद से करो. Take your time, take your 30 seconds and try to solve this. कर सकते हो, कोई बहुत क्वेश्चन डिफिकल्ट ये है नहीं, आराम से किया जा सकता है. Identify the correct statement with regard to the G1 phase of the interphase. Aditya has already raised his hand. Okay, Shikha, बताइए.
Sir, option C.
Shikha is saying here option C. All right, Gayatri.
Sir, option A.
Gayatri, option A.
Yes, sir.
चलो गायत्री को भी हम देते हैं यहाँ पे हमारे मार्क्स. Remember this, Gayatri. From next test, 25 marks will be deducted.
Sir, but in X phase only the DNA replication take place.
लेकिन G1 phase लिखा है रे बाबा. Gap 1 phase, Gayatri. What are you saying?
Sir, in S phase only DNA replication take, but in D1 phase not.
Wait. अच्छा, Aditya.
Sir, it's C, sir.
See, all right. Who else is remaining right now? सबने दिया ना? महालक्ष्मी का आंसर आ गया ना? महालक्ष्मी का तो.
Yes, अभी आ गया sir, option C.
ठीक है. So it is going to be Shikha का भी आ गया ना? Shikha, it's C. Ashika is already.
Yes, sir.
So it is your option C in this. Right. See, now they have asked you which of these is going to be a correct statement with regard to the gap 1 phase. With regard to gap 1 phase, G1 phase of your interface, which is a correct statement. Statement A क्या करा मैंने यहाँ पे DNA synthesis और replication takes place. That's a wrong statement. ये G1 phase में नहीं होता, ये होता है S phase के अंदर. ठीक है? हाँ.
So I thought
Reorganize.
incorrect statements.
Yeah, tell me
Sorry, sir.
Gayatri. बोलो बोलो.
Sir, I thought incorrect statements, sir. Thought reading the question properly. Sorry.
See, now you have minus 25. Now you have a depth of minus 25. काटते रहना अगले टेस्ट में से जो भी स्कोर आएगा धीरे धीरे. Alright. Reorganization of all the cell components takes place. That's also not true. That is not something will be happening in the G1 phase, right? Nuclear division takes place. किसने बोला? किसने नहीं बोला? So that's also untrue. Option C becomes my correct answer. Cell is metabolically active, grows but does not replicate its DNA. So yes. So NEET 2020 का क्वेश्चन ये तो हम लोग कर चुके हैं, इसको वापस नहीं करना, ये 2019 में आ चुका है, राइट? ये भी हम लोग कर चुके हैं. ऑप्शन ए becomes my correct answer here. During cell growth, DNA synthesis takes place on this phase, राइट? हाँ, अब मैं इसके अंदर आपको थोड़ी हेल्प कर दूंगा बिकॉज ये स्टेटमेंट आपको यहाँ पे समझ नहीं आएगा बिकॉज ये आपको 12th में आता है. When the cell has a stalled DNA replication fork, I would rather help you out in making this easy. Stalled DNA replication fork can be taken right now as a, let's say you have a error in the DNA. You have an error in DNA. इसको मैं एक्सप्लेन कर कैसे एक सिंपल लैंग्वेज में आपको ये दे रहा हूँ यहाँ पे अभी, राइट? क्योंकि ये स्टॉल्ड रेप्लिकेशन फोक आप 12th में सीखते हो, इसका मतलब क्या होता है. Okay, so assume that you have an error in the DNA. Which checkpoint should be predominantly activated? Now you have to tell me.
Answer B.
Aisha is saying, Aisha, you have to raise your hand. Huh? You have to raise your hand. You don't raise your hand, Aisha. Aisha is from Mangalore, right? Aisha, how's temperature in Mangalore, by the way?
Lol.
It's
Hi, sir.
It's high. Okay. All right. Yeah, Aditya, tell me what will be the answer over here.
Sir, A.
A. Aditya. Mantasha is.
Sir,
Option
B,
B.
sir. B, sir.
हो गया अब हो गया. Mantasha is like option B and Gayatri, Gayatri, tell me. Gayatri is saying nothing. Shikha, क्या होगा इसका आंसर?
Sir, option B.
Option B is going to be a correct answer. Yes, if you have error in your DNA, like which I have given you a hint over here, if you have a DNA error, some issue with the DNA, then you will find checkpoint number B. That means your checkpoint number G2 or M checkpoint. That is going to be checkpoint number 2 will be my active. This checkpoint has to be active over there. Okay? So that is going to be my option B over here. Right? Now, in S phase of the cell cycle, which of these events will take place? Take your time, take your 30 seconds and then try to tell me the answer over here. Some people have raised their hand. शिखा ने हैंड रेज कर दिया है. शिखा बताइए क्या होगा इसका आंसर?
Sir, option A.
शिखा इज सेइंग ऑप्शन ए. ऑलराइट, बाकी लोग को हम लोग टाइम देते हैं यहाँ पे. महालक्ष्मी, टेल मी.
Sir, option A.
ऑलराइट, एंड आयशा.
Yes, sir.
गायत्री.
Sir, option A.
Option A is going to be correct. I guess Aditya and all the other people over here will also agree with this. Option is going to be correct answer. Amount of DNA doubles in each cell. So in the S phase, you will find the amount of DNA is going to get doubled over here, right? So that's going to be a true statement. बाकी सब यहाँ पे गलत है. बाकी statement आपके यहाँ पे amount of DNA remains the same in each cell. That's going to be a wrong. Chromosome number is going to increase. That's going to be wrong over here. So yes, option A becomes my correct answer. Right now you know all those small things, right? Now this was a question they have asked in the कल के एग्जाम के अंदर. इसे तुम लोग कर सकते हो. सिंपल है, बहुत सिंपल है. Take your time, take your 20 seconds and let's see 2024 के नीट का क्वेश्चन आपसे हो रहा है नहीं हो रहा. होना ही चाहिए obviously. 158 A B C D E. You have to rearrange them, right? So you can actually do this question. कर लो, चलो करो और हैंड रेस करो अपना. Aditya, tell me.
Sir, fourth one, sir.
Aditya is saying option 4 over here. Shikha.
Sir, option D.
Option D. All right. Misbah. Oh, Misbah is there in the class.
Option 4.
Misbah आपको भूल गए हम. ठीक है, option 4. All right, and all of you are giving me option 4 over here. I think all the remaining students over here will give me option 4, I guess. So yes, that's going to be a correct answer, right? So first going to be my gap 1 phase, then we'll have a gap 2 phase. इतना भी मालूम हो तब भी आपको आंसर मिल जाता है. Only if you know this initial sequence, you get your answer, right? Then you have your synthesis phase, right? That is something you'll get to see. Okay, एक second. Yeah, sorry, yeah. E, C, and A, right? So that's something also you get to know. You will get to know that you will have answers. So E, C, A, and then you have your karyokinesis, and then you have a cytokinesis. So this is my option 4. Right. This is something we can do later. अभी हम पढ़ते थोड़ा आगे. Now what we'll discuss right now, we'll discuss something in terms of the mitosis. Now mitosis, what will happen? Mitosis will have 4 phases. Now you know those 4 phases. You have a prophase. Someone has a question क्या? Is the hand raised क्या बताओ? सवाल है तो बोलो. No question. Right. Now, when we discuss a mitosis, you know that mitosis will have the 4 phases. You will have your prophase, you will have your metaphase, you will have your
anaphase.
Anaphase.
and then you have a telophase.
Telophase.
Something you already all of you know, right? So I really don't have to repeat those things. We'll start with the prophase. So let's jump onto the story of my prophase and what are the things I'll be finding happening in my prophase. Now, when you observe the prophase, what are the things you get to see happening? Now, I'll make a large nucleus, right? मैं cell बना रहा हूँ, मैं nucleus बना रहा हूँ यहाँ पे direct. So you have a large nucleus, and what are the events which are happening in the nucleus? Let's try to understand those things. So you will find the nuclear membrane will start breaking or will disassemble in the prophase. But that is not the first thing which is happening. ये पहली चीज यहाँ पे नहीं हो रही. सबसे पहले पता है क्या होगा prophase के अंदर? You will find your DNA will undergo condensation to form the chromosome. So the DNA you have or the chromatin you have, that will undergo condensation and you will find chromosome-like structure will start forming over here.
Chromatin network.
Yes, chromatin will get condensed to form the chromosome. So that is the first thing you will get to realize happening over here, that your chromatin condense to form chromosome. So basically condensation of chromatin initiates. Condensation of chromatin is going to start and will get completed, right? I won't say get completed completely, but it will happen in the prophase. So that is something you will get to see. So chromatin condensation of chromosome, that will start happening in my prophase. That's the first thing you have to know. Second thing you have to know that my nuclear membrane will disassemble. My nuclear membrane will disassemble. That means it will start breaking. So my nuclear membrane will start breaking over there because that is also one of the event of my prophase, right? That my nuclear membrane will start breaking because by the end of the prophase, you don't have a nucleus. Once the new prophase gets completed, you don't get to see a nucleus over there. So nuclear membrane will start to disassemble in the prophase. That's the second thing you'll find happening. The third thing you will find happening over here is your centrioles will start moving towards the poles, right? So assume that you have your centrioles. So now let's say you have one centriole over here, which is kept inside the centrosome. ठीक है, ये मेरा एक यहाँ पे सेंट्रियोल है और एक मेरा यहाँ पे है. दोनों अपोजिट जाना चालू करेंगे. Both of them will start moving apart from each other. So you have one centriole over here, one on the opposite side, and both of them will start moving towards the poles. Right? So you have the centrioles over here. You have So now what will happen? They will start going to the pole. So this will go in one particular direction, and this will go in one particular direction. So they start moving towards the poles. They move towards the opposite poles, right? So that is one thing you have to know, that centrioles will start moving towards the poles. Centrioles starts moving towards poles. And along with that, there's one more thing which is happening with them. These centrioles will start forming my spindle fiber. So spindle fiber formation will initiate. तो ये जो आपके सेंट्रियोल्स है ना, जो आपके पोल्स की तरफ आपके जा रहे हैं, उनके अंदर से स्पिंडल फाइबर फॉर्मेशन आपका इनिशिएट हो जाएगा. So you will find the spindle fiber formation will initiate from them. तो यहाँ से आपका स्पिंडल फाइबर फॉर्मेशन आपका होने लगता है. So that will also keep on happening over. So ये भी याद रखना, that spindle fiber formation will initiate in this particular stage. So now let me remove this on the right hand side, the space I have taken. Now let me just remove this, because that is not something you already know this. So you will find. हाँ जी बेटा बताइए, महालक्ष्मी बोलिए.
स्पिंडल फाइबर्स और क्रोमोसोम ऑपोजिट पोल में जाते हैं ना सर, वो मेटाफेज है ना सर?
हाँ, लेकिन यहाँ पे क्रोमोसोम थोड़ी जा रहे हैं, यहाँ पे तो सेंट्रियोल जा रहे हैं. हाँ, वो मेटाफेज.
सेंट्रियल जाने वक्त स्पिंडल फाइबर कैसे फॉर्म होगा सर?
देखो,
वो मेटाफेज में होगा ना सर?
ना, मेटाफेज में तो आप क्रोमोसोम को पकड़ के बीच में लेके आते हो. मेटाफेज के अंदर,
स्पिंडल फाइबर.
हाँ, विद हेल्प ऑफ द स्पिंडल फाइबर. अभी आपने बस चालू कर दिया, you have just initiated the formation of the spindle fiber, right? We are not saying the spindle fiber will go and attach with the chromosome and will bring them in the center. You have just started the initiation of the spindle fiber, and that will keep on happening as this centriole will start moving towards the poles. तो जैसे मेरे यहाँ पे centriole poles की तरफ जाना चालू करते हैं, at the same time I will be finding my spindle fiber formation will also initiate. तो ये याद रखना, ठीक है? So you will find. महालक्ष्मी समझ आ रहा है?
हाँ सर, एक बार ड्रॉ करके
देखो, ड्रॉ तो यहाँ पे क्या है कि ये जो आपका निकल के आ रहा है ना, यहाँ पे आपका ये आपका स्पिंडल फाइबर आपका यहाँ पे निकल के आ रहा है. ठीक है,
सेंट्रियोल्स में से सर.
हाँ, सेंट्रियोल्स में से ये जो टी बना हुआ है ना, टी दैट इज माय सेंट्रियोल्स, इसमें से स्पिंडल फाइबर आपका निकल के यहाँ पे बाहर आ रहा है, राइट? और ये जो मेरे सेंट्रियोल्स है, दोनों अपोजिट पोल्स की तरफ जाना चालू करेंगे यहाँ पे. सो याद रखना, स्पिंडल फॉर्मेशन आप सिंपली यहाँ पे लिख सकते हो, आप यहाँ पे स्पिंडल फॉर्मेशन इनिशिएट्स. That means the spindle fiber formation is going to start right now, right? It will just initiate. So these are the things you have to know. Spindle fiber formation will initiate. Centrioles में से ये बन रहा है. Okay, this is coming from the centrioles, and centrioles are moving towards the poles. Along with that, chromatin will get condensed from chromosome, and nuclear membrane will disassemble. These are the 4 things you have to know happening in my prophase. ये 4 चीजें मेरी हो रही होती है मेरे यहाँ पे प्रोफेज के अंदर. आयशा समझ आ रहा है यहाँ तक? आयशा, is this clear to you?
Yes, sir.
Okay. Yeah, and now this question goes to all of you over here, right? I hope this is clear to all of you. These are the 4 things which I'll be finding happening over here. Misbah, Shikha, Gayatri, Aditya,
Yes, sir.
Mahalakshmi, and Asha. Okay? And I hope I'm not missing anything, right? So these are the 4 things you will be finding happening in the prophase. Okay, now you will get to see that during the prophase, as you can see, my DNA will get condensed and will turn into the chromosome. You have an early prophase, and by the end of the late prophase, you will find the nuclear membrane is almost gone. आपकी न्यूक्लियर मेम्ब्रेन काफी जा चुकी है और ये अभी और भी निकल जाएगी, राइट? जब तक प्रोफेज एकदम खत्म होगा तो न्यूक्लियर मेम्ब्रेन गायब, एंड यू कैन सी द क्रोमोसोम्स ओवर हियर. यू कैन सी द कंडेंस्ड क्रोमोसोम. कैन सी द कंडेंस्ड क्रोमोसोम. The best way to see this happening ना in the onion peel. आपके स्कूल के लैब में जाओ, मैडम को बोलो कि चाबी चाहिए लैब की, अंदर जाओ और अनियन लेके जाना, एंड अनियन का पील निकालो, बढ़िया स्लाइड पे लगाओ, डालो उसके ऊपर आपका मिथिलिन ब्लू डालो उसके ऊपर, राइट? अलाउ इट टू स्टेन फॉर 30 seconds and then observe the onion peel, and you will find some cells will be undergoing the mitosis. So some will be in the prophase, some will be in the metaphase. बल्कि metaphase, anaphase काफी clearly दिख जाता है. Metaphase, anaphase, and even the telophase is very something which is clearly seen, right? If you are paying good attention. So do that. Go to your school and do it by yourself. You are the PCB students. तुम्हारे पास एक्सेस नहीं होगा लैब का तो किसके पास होगा? किंडरगार्टन के पास. So this is something you have to know. right? So this is what you find happening in the prophase. Now the next thing which will be happening is when we have metaphase. Now we will discuss the metaphase. अब इसका जो आपका next stage आपके यहाँ पे आता है, that is going to be the metaphase. Now metaphase is a time where we'll get to see this is the best time to study chromosomes. ये आपका सबसे बढ़िया टाइम होता है क्रोमोसोम स्टडी करने का, whether it's an animal cell or a plant cell. Metaphase is the best time to study chromosome because in this stage what will be happening, all the chromosomes will be aligned in the center. All the chromosomes will be aligned in the center. So let's say this is one chromosome. Wait a minute. This generally happens, not double lining, because of the sweat. So yeah, you have this chromosome over here, then let me draw a few more chromosomes. Right. So these all chromosomes are in the center and they have been brought in the center with the help of my spindle fibers. And those spindle fibers have been actually coming from my this particular centriole. This spindle fiber are actually coming from all those my centrioles. So ये centriole से मेरे यहाँ पे spindle fiber यहाँ पे आ रहे हैं और इनकी वजह से ही जो centrioles मेरे pole पे जा चुके थे, they have started moving towards the poles in the prophase. Now they have reached towards the poles and from there what they are actually doing over here, they are actually aligning the chromosomes in the center, right? Now this is something you have done in your 11th grade, right? This is not something which is going to be new to you. You have seen this thing happening in the 11th grade. So they are going to be bringing them into the center. Now the question is कि center में ला कैसे रहे? How they bring them into the center? Because this spindle fiber will get themselves attached to a particular area in the chromosome.
Kinetochore.
Yes, that area is called kinetochore. बिल्कुल सही. We have seen this thing. We have seen kinetochore present in the chromosome. So these spindle fiber are getting themselves attached to the kinetochore, and because of that, they are able to bring them into the center, which is one of the important event of my metaphase. So all the chromosomes are in the equatorial plate. They are all in the center. So we say that they have been brought in the equatorial plate. So let's see, they have been brought in the equatorial plate. सेंटर में लाके खड़ा कर दिया, इक्वेटर भी सेंटर में ही होता है, so that is why we use this term. That is what you will be finding happening in the metaphase. That's what you have to know. So if they ask you, generally they are going to ask you this question, that which phase of the mitosis is the best time to study the chromosome, the answer is going to be my metaphase. At this point you can actually see all my chromosomes in the center. You can actually study them, you can count them, you can see if there is some kind of deformity in the chromosome. कई बार होता है क्रोमोसोम के अंदर भी आपको डिफॉर्मिटी मिल जाती है, क्रोमोसोम का कोई एक हिस्सा टूटा हुआ होता है, so you find that happening, so you can see the deformity in this particular stage. Sometimes happens that a cell should have some number of chromosome and it is not there, it is missing, तो भी आप यहाँ पे check कर सकते हो. So it is the best time to study my chromosome. So याद रखना, metaphase is the best time to study my chromosome. So this is again should be clear to all of you, right? Now, so once we are done with this, now after this we'll be having, so you can see over here, this is my metaphase. You can get to see your all the chromosomes are aligned in the center. Center में लाके हमने इसे खड़ा कर दिया. So that is all happening in my metaphase, right? I need some water. No water nearby. All right. So now you will have, after this, you will be having my next phase. That is my anaphase. ये मेरा यहाँ पे हो गया. अब मेरा यहाँ पे आ गया anaphase. अब anaphase आने के पहले अब यहाँ पे हमें कहानी समझते हैं. अब हम बात करेंगे हमारे यहाँ पे चेक पॉइंट नंबर 3 के बारे में. Now over here we'll discuss about the check point. जो आपने नहीं हमने बोला था check point नंबर 3 आपका आता है माइटोसिस के अंदर, M phase के अंदर. अब हम इस बारे में यहाँ पे बात करेंगे. So check point number 3 is going to be between my metaphase and anaphase. याद रखना, metaphase और anaphase के बीच में आपका यहाँ पे होता है आपका चेक पॉइंट नंबर 3. ये बात आप अपने दिमाग में बिठा लो, ठीक है? अब समझते कि यहाँ पे होता क्या है. सी, जनरली होता क्या है, नाउ वी अज्यूम दैट यू हैव अ क्रोमोसोम्स ओवर हियर इन द सेंटर इन द मेटाफेज, यू हैव दिस क्रोमोसोम्स इन द सेंटर इन द मेटाफेज, राइट? दो बनाते हैं, एक से काम नहीं चलता। इनको सेंटर में कौन लेके आया?
कैनेटोकोर,
अरे नहीं, स्पिंडल फाइबर। कैनेटोकोर तो आपका वो हुक होता है,
स्पिंडल फाइबर्स।
स्पिंडल वेब,
स्पिंडल फाइबर इनको सेंटर में यहाँ पे लेके आ चुके हैं, राइट? और स्पिंडल फाइबर अटैच थे मेरे किसके साथ?
कैनेटोकोर,
कैनेटोकोर के।
कैनेटोकोर्स।
ठीक है, अब एक सिचुएशन को तुम यहाँ पे इमेजिन करो, राइट? हमने इन्हें यहाँ पे बीच में लेके आए हैं और अब यहाँ पे आपका होगा एनाफेज। एनाफेज में क्या होता है? बेसिकली आपका सेंट्रोमियर ब्रेक होगा, right? The centromere is going to break and my two chromatids will start moving apart. So in the, so what you get to see happening in the anaphase, that your centromere is going to break, and after the breaking of centromere, what will be happening? That your one chromatid will move towards one pole, other chromatid will move towards the another pole. So that is what you find happening in the anaphase. एक सक्सेसफुल एनाफेज के अंदर आपको ये चीज आपको होते हुए आपको मिलती है कि एक क्रोमैटिड ऊपर जाएगा और दूसरा क्रोमैटिड आपका नीचे जाएगा. So that is what you get to see happening, ना? That is what you find generally happening in my एनाफेज. सक्सेसफुली यही होता है एनाफेज के अंदर कि ऐसे खींच के आपके क्रोमैटिड को आप अलग कर लेते हो. ये बात आपने 11th में भी देखी थी और आपको ये बात हमेशा आपको याद भी रखनी है. दैट एनाफेज मतलब अपार्ट. The chromatid will move apart, right? So this will go in this direction and this will go in this direction. That is what is happening in the anaphase. Now let's understand a story over here. Assume that Gayatri and Mahalakshmi are holding on to a pumpkin. You know na pumpkin. Pumpkin is what do you call
Yes,
pumpkin in
sir.
Hindi? Misbah, Hindi में क्या बोलते हैं यार? पुmpkin को भूल गया.
कद्दू।
Kaddu, right? Sorry.
I don't know.
Kaddu, right? Okay. So yeah, you have a pumpkin and there is a sabji you make for a pumpkin, right? Pumpkin की सब्जी बनाते हैं हम लोग, ठीक है? Now, so assume that Gayatri is standing on one side, Mahalakshmi is standing on one side, right? And they have a pumpkin with them, and they don't have a knife to cut the pumpkin into half. उनके पास knife नहीं है pumpkin को दो half में काटने के लिए. तो महालक्ष्मी comes with a plan कि इसको हम रस्सी से बांधते हैं, right? और गायत्री तुम वहां से खींचो, मैं यहाँ से खींचती हूँ. So we'll pull the pumpkin apart, दोनों साइड से बांध के, तो वो बीच में से टूट जाएगा. And then we can take those pumpkin घर पे, right? So उन्होंने रस्सी से बांधे वहां पे. हाँ महालक्ष्मी बताओ, बोलो महालक्ष्मी बोलो.
Sir, best example is balloon.
बलून? फाइव बलून?
The furrow formation होगा ना sir.
अच्छा छोड़, बलून हटो, हमें pumpkin की चाहिए. Right? Now you tell me. So now you have a Mahalakshmi holding on to a pumpkin with the rope from one side. From one side is going to be Gayatri. And now what actually happens over here is that both of them will start pulling. Now, when they start pulling, now imagine there is a situation that Gayatri is not holding on to rope tightly. Gayatri अगर rope को tightly पकड़ के नहीं रखेगी, तो मुझे एक बात बताओ, suddenly क्या होगा? What will happen suddenly?
Unequal group.
Pumpkin will go towards the Mahalakshmi side. वो ब्रेक नहीं होगा. ब्रेक तब होता है जब आप दोनों से एक साइड से अगर आप खींच रहे होते. तो गायत्री ने तो उसे पकड़ के नहीं रखा था, गायत्री ने उसे काफी लाइटली पकड़ के रखा था. तो अगर कोई लाइटली पकड़ के रख रहा है, then what will happen? It won't break from the center, ना? It won't break. So what will happen? The complete pumpkin will go onto the Mahalakshmi side. मेरी बात समझ आ रही है आपको? यहाँ पे क्या हुआ?
Yes,
आपने
sir.
उसे ब्रेक
Yes,
करने के लिए
sir.
दोनों साइड से उसे खींचा. You are trying to break that pumpkin, so that's why you are pulling them from the both the sides. But because one of the person was not holding that particular pumpkin strongly, what actually happened? The pumpkin did not break, and the complete pumpkin went onto one side on the Mahalakshmi side, क्योंकि वहां से वो खींच रही थी. ठीक है? अगर ऐसा हुआ तो इसका मतलब क्या हुआ? इक्वल डिस्ट्रीब्यूशन आपका हुआ ही नहीं यहाँ पे. So that means what? There was no equal distribution taking place over there. क्या ये वाली बात आपको यहाँ पे समझ आई?
Yes, sir.
Yes, sir.
Right?
Yes, sir.
I hope Gayatri is clear to you as well, right? And Aditya, you as well, and Misbah, you as well, right? So now you don't have equal distribution of pumpkin happening over there. So complete pumpkin went on to one particular side. Same thing can happen over there. सेम चीज आपके यहाँ पे हो सकती है अगर आपके स्पिंडल फाइबर सही से यहाँ पे अटैच नहीं है तो. अब तुम सोचो कि अगर तुम्हारा स्पिंडल फाइबर ये जो ऊपर से आ रहा है ये यहाँ पे सही से अटैच यहाँ पे नहीं है, ये लूजली अटैच है, इसने इसे टाइट से नहीं पकड़ा. द स्पिंडल फाइबर इज नॉट टाइटली अटैच्ड टू माय कैनेटोकोर. अगर ऐसा होता है तो यहाँ पे क्या होगा? द कंप्लीट क्रोमोसोम विल गो इन वन डायरेक्शन, इट वोंट स्प्लिट. द सेंट्रोमियर विल नॉट स्प्लिट. आपका सेंट्रोमियर यहाँ पे स्प्लिट हो क्योंकि यहाँ पे आपका वीकली अटैच है. अगर ये आपका यहाँ पे वीकली अटैच है आपका यहाँ पे, तो जब खींचा जाएगा, जब दोनों साइड से आप खींचोगे तो क्या होगा, ये पूरा क्रोमोसोम आपका नीचे चला जाएगा। क्या मेरी बात आपको यहाँ पे समझ आ रही है? इज दिस क्लियर टू ऑल ऑफ यू?
Yes, sir.
The complete chromosome will go in one particular direction. I hope Gayatri it's clear to you as well, because I'm speaking Hindi in somewhere in between. That's why I'm asking you again. Gayatri, you were saying something. हाँ ठीक है. So if you keep this attached over here weakly, then what will happen during the pulling of this from the two sides? What will happen? The complete chromosome will move in one direction. So that is what will happen. Na, ये पूरा chromosome, this complete chromosome will go in one direction, and that is not a good thing. By no means that is a good thing. If at all that happens, na, that's not a good thing, because in that case what will happen, the cell will not get one chromosome. One cell will not have one chromosome and another cell will have one extra chromosome. This is not an equal distribution, this is an unequal distribution. So that can lead to a unequal distribution. This will lead to an unequal distribution, and this is not something you want. So that is why you have to ensure that आपको ध्यान रखना है that my all spindle fiber are tightly attached to the kinetochore. आपको ये ध्यान रखना होता है कि आपके सारे स्पिंडल फाइबर kinetochore से सही से अटैच है. अगर वो अटैच नहीं होंगे तो during the pulling of the spindle fiber, there is a likely chance that one side will get complete chromosome and other side will not get anything. So that is why checkpoint number 3 will come into picture. Checkpoint number 3 will have a role to ensure that it checks the attachment, or check the attachment of, it is going to check the attachment of spindle fiber with kinetochore. That is the first thing it is going to check. ये पहले वो चेक करेगा यहाँ पे कि ये सही से अटैच है नहीं अटैच है. अगर वहां पे वो सही से अटैच है, अगर लग रहा है उसे कि वहां पे सब कुछ सही से अटैच है, then there is one complex, there is one protein complex called as my APC, anaphase promoting complex. लिख लेना ये चीज. APC stand for my anaphase promoting complex. That is also becomes a part of my checkpoint number 3 only, anaphase promoting complex. तो पहले आप चेक करोगे क्या कि आपके लगे हुए वहां पे सही से अटैच है नहीं अटैच है. Once you have seen that okay, my spindle fiber are strongly attached to my kinetochore, after that you will have the anaphase promoting complex, and this anaphase promoting complex will trigger the promotion of the anaphase. So अब वो क्या बोलेगा कि चलो अब anaphase चालू करते हैं. हमने देख लिया सब कुछ वहां पे सही से अटैच है. अगर वहां पे सब कुछ सही से अटैच है तो आप क्या करते हो, यू इनिशिएट द एनाफेज. और वो इनिशिएट कौन करेगा, मेरे कुछ प्रोटीन यहाँ पे, और उन प्रोटीन को हम बोलते हैं यहाँ पे एज अ एनाफेज प्रोमोटिंग कॉम्प्लेक्स. सो विद द हेल्प ऑफ देम योर क्रोमैटिड सेग्रीगेशन और क्रोमैटिड सेपरेशन विल हैपन सक्सेसफुली. इसकी वजह से आपका एनाफेज आपका सक्सेसफुली हो पाता है. तो ये बात आपको यहाँ पे याद रखनी है. दिस शुड बी क्लियर टू ऑल ऑफ यू. तो क्या ये वाली बात सबको क्लियर है यहाँ पे? आदित्य, मंताशा, गायत्री, शिखा, मिसबा, इज इट क्लियर टू ऑल ऑफ यू?
Yes, sir.
Yes, sir.
Sir, can you repeat once for the APC?
So APC stand for my anaphase promoting complex. Its role is going to be to promote the anaphase. Once you have seen that my all spindle fiber are tightly attached to my kinetochore, then what you have to do, you have to give the signal. You have to tell कि चलो अब करते हैं anaphase. So चलो करते हैं anaphase कौन बोलेगा? वो बोलेगा ये anaphase promoting complex. Now it will tell okay, now let's initiate the anaphase. And after this you will find my centromere will break and my both the chromatid will start moving apart. So this is what my anaphase promoting complex is known for. It will promote, it will initiate the anaphase. Okay, so please remember, if the APC is not working, if the anaphase promoting complex for some reason is not working properly, then the anaphase will not be taking place over there. ठीक है, तो ये बात आपको यहाँ पे याद रखनी है, right? So now this should be clear to all of you. Now let me, so this should be clear to you. This is what you will be finding happening over here. Okay.
Yes,
Now let me
sir.
show you one question over here. I don't have a question. I have a question with the answer. I don't want to give you that question. All right. So I will give you a couple of questions in the homework. Now you have to answer that in the next class, right? So this is what you will be finding happening over here. This is something you should remember happening over here. Now if I see the anaphase now, so now you can see what has been happening in the anaphase. हम यहाँ पे देख लेते हैं. So anaphase के अंदर यहाँ पे मेरे chromatids जो होंगे, all the chromatids will start moving towards the pole. So centromere has broken and the chromatid have started their journey towards the poles and they have been pulled up by my spindle fiber. इनको खींच के ले जा रहा है मेरे यहाँ पे spindle fiber. वो भी बना लेते हम लोग यहाँ पे. So ये spindle fiber इसको खींच के ले जा रहा है. अच्छा, by the way, all of you have given the test ना? कोई था आप में से जिन्होंने टेस्ट नहीं दिया? आदित्य ने टेस्ट नहीं दिया. Why आदित्य?
Sir, I was absent from past 1 week, sir.
Yeah, you were absent from past 1 week. Yes, like you were not there in the like you were traveling somewhere.
Yes, sir. I was traveling.
Okay. ठीक है. Just try to ensure now. चलो अभी तो चलो 11 का bridge course चल रहा है, but at least from 12th onwards, make sure that you don't miss out of the classes.
Yes, sir. I won't miss classes.
So this is something you will be finding over here that this is my anaphase. So my chromatid are moving apart, right? And they've been pulled apart by my spindle fiber. So this is what you find happening in my anaphase. ठीक है? ये चीज आपको anaphase में होते हुए मिल जाएगी यहाँ पे आपको. Anaphase is a very simple phase. Doesn't take much amount of time. Complex भी नहीं है ये ज़्यादा. खींचो और अलग कर लो इन्हें. Right? This is what you find happening over here. Okay. So after this, you will be having your telophase. Now, telophase is something we'll have a look at that later. Apparently, I want all of you to remember this thing because next time when we'll meet, we'll do the telophase and then we'll jump onto the meiosis. Right? We'll solve some questions and then we'll jump onto the meiosis. That's something we'll be doing over here, and I'll give you some questions over here as a homework as well. Right? So that is what we have done. So in today's class, we have done the initiation of the cell cycle. What happens in cell cycle? What is my cell division and everything over there. And then we have moved on to something like your mitosis. We are done with the prophase, we are done with the metaphase, and we are also done with the anaphase. And we have also seen all the 3 checkpoints. Checkpoints, yaad rakhna, we have seen question coming from checkpoint in the past. So there's a likelihood that they might come again in the future as well. Right? Kal nahi aaye the, but aapka exam to 25 mein hai, to kya pata aa jaye. So please remember what are these checkpoints and you should know what they are actually are known for. ठीक है. अच्छा, ये क्वेश्चन कर दो. ये क्वेश्चन भी कर सकते हो. कल का ही क्वेश्चन नहीं है, मैंने यहाँ पे लगा के रखा था. सेल द यूनिट ऑफ लाइट तो आप लोग पढ़ चुके मेरे साथ और स्कूल में पढ़ाई आपने. सो टेल मी व्हाट इज गोइंग टू बी करेक्ट मैच फॉलोइंग ओवर हियर. रेज योर हैंड टू आंसर दिस. ऑलराइट शिखा, टेल अस व्हाट इज गोइंग टू बी करेक्ट आंसर.
Sir, option A.
शिखा इज सेइंग ऑप्शन ए. पक्का? श्योर?
Yes, sir.
शिखा, क्या लिखा हुआ है मैंने यहाँ पे? बोलो.
Minus 25.
हाँ, ठीक है. अगले टेस्ट से काटेंगे हम आपके. राइट? अगले टेस्ट में if you get 120, then your school we will may bring it down to 95. ठीक है. चलो शिखा, -25. Anyone else? महालक्ष्मी बताइए.
Sir, option B, 2.
ऑप्शन बी. महालक्ष्मी ऑप्शन बी करें यहाँ पे. Someone else also has raised their hand. Misbah, tell us. Shikha has only raised her hand again. Achha. Anyone else? No one else. That's it. Gayatri, Misbah, Aditya. बस इतना ही. एक ही पर्सन. अरे बाबा, we have done this ना. सिट्रिक एसिड साइकिल मतलब क्या होता है? Tell me something. सिट्रिक एसिड साइकिल आपका क्या होता है? It is a Krebs cycle. याद आ रहा है?
Yes, sir.
सिट्रिक एसिड साइकिल इज अ Krebs cycle, right? और Krebs cycle कहाँ पे होता है? आप बताओ मुझे. Krebs cycle होता क्या है? 1 2 3 4
Mitochondrial matrix.
Mitochondria.
मैट्रिक्स. हाँ, माइटोकॉन्ड्रियल मैट्रिक्स यहाँ पे. सो ए के साथ 2 लगेगा यहाँ पे. अब ए के साथ 2 सिर्फ एक ही ऑप्शन में आ रहा है यहाँ पे. ए के साथ 2 एक ही ऑप्शन में आपके यहाँ पे आ रहा है, राइट?
Yes, sir. Option B.
You should know, Baba. Glycolysis will always happen in the cytoplasm. Glycolysis will also happen in the cytoplasm all the time. इलेक्ट्रॉन ट्रांसपोर्ट चेन तो हमने पढ़ा था ना थोड़े दिन पहले. ईटीसी कहाँ पे हो रहा होता है? Inner membrane of
Inner
body.
membrane.
How can you like forget something so early, Baba? So yes. So C के साथ मेरे यहाँ पे 4 आएगा. So आप देख लो, A के साथ 2, C के साथ 4 मेरे यहाँ पे है. C के साथ 4 भी ऑप्शन 2 में ही आ रहा है सिर्फ. कितना यूनिक है यहाँ पे. तो आपको काफी आसानी से यहाँ पे आंसर मिल जाना चाहिए था. ऐसे क्वेश्चन आपको सॉल्व करना सीखना पड़ेगा. You cannot make such a, if in the match of following, if you know 2 things, no, you can get your answer. most of the time. So option 2 become over here my as a correct answer. Okay. All right, then chalo. So that's it from my end. If you now have any questions from your side, if you have any questions from your end, then please go ahead and ask your questions. अगर आपके कोई सवाल है यहाँ पे, then अपने सवाल आप यहाँ पे पूछ सकते हो. If you have no question, then I'll be sharing with you the slides and some questions as a homework as well. So you can solve those questions घर पे बैठ के.